Okay, imagine this. A plant. It lives in these really acidic bogs, right?
Hmm?
And it doesn't get its food from the soil, not really. Instead, it actually catches insects and digests them. The sun do.
Oh yeah, a carnivorous plant. Darwin himself was absolutely fascinated by them.
Exactly. And it makes you wonder, how does something so specific, such a unique adaptation, actually come about?
Hmm.
And not just that, but, all the incredible mind-boggling diversity of life we see everywhere.
It's the fundamental question, really.
So in this deep dive, we're kind of taking a shortcut to unpack one of biology's biggest ideas, the process of evolution. We're using Chapter 11, The Process of Evolution, from the textbook Raven Biology of Plants.
Mm.
Our goal here is simple. Pull out the key ideas, explain the tricky bits in plain English, and really connect the dots for you.
So you can get the core mechanics of how life changes, yeah, without needing complex diagrams right in front of you.
Exactly. We'll start right at the beginning with Darwin, and then move through population genetics, how new species form, and the bigger evolutionary patterns.
Sounds like a plan.
Okay, so before Darwin, the dominant idea was special creation. The belief that every living thing just appeared, boom, in its current form, fixed.
Right. Though there were some early evolutionary thinkers like Lamarck, Jean-Baptiste Lamarck, but his ideas about how change happened, like inheritance of acquired characteristics, they just didn't quite hold up.
Right. Then along comes Charles Darwin. He's just 22, joins the HMS Beagle voyage in 1831, 5 years around the world. And his book, The Voyage of the Beagle, it's more than just a travel journal, isn't it?
Oh, absolutely. It's like reading his thought process develop. You see the observations piling up, the questions forming.
And the Galápagos Islands, they were just pivotal.
A natural laboratory.
You saw plants, animals there, distinct from anywhere else, but weirdly similar to those on mainland South America nearby.
Which immediately raised a flag for him. If everything was specially created for its location, why the resemblance to the mainland forms?
And even more, the locals could tell which island a giant tortoise came from just by its shell shape.
Yeah. If they were all created separately, shouldn't they be, well, identical? Or at least not patterned by island?
So Darwin started wondering, could these island creatures have actually come from mainland ancestors?
Mm.
Maybe they spread out, colonized the islands, and then slowly changed, adapted to fit each specific island environment.
It's such a powerful line of reasoning. And he wasn't entirely alone in this thinking. Influences were there. Thomas Malthus's essay from 1798, the one about population growth potentially outstripping food supply, that was huge for Darwin.
How so?
Well, Darwin realized Malthus's struggle for existence wasn't just about humans. It applied to all life. Most populations don't grow exponentially forever. Something limits them. Death, resource scarcity.
Okay.
So if there's variation within a population, which Darwin clearly saw, and there's this struggle, doesn't it follow that individuals with variations that give them any edge are more likely to survive?
And reproduce.
And reproduce.
Yeah.
Passing those advantageous traits on, that was the core of natural selection.
And Alfred Russel Wallace had the same idea, independently.
He did. Which kind of pushed Darwin to finally get his big book out on the origin of species in 1859.
He used artificial selection as an analogy, right? Like with
Yeah.
dog breeds or plants.
Exactly. Think about wild mustard, Brassica oleracea. Just look at what human breeders did with that one species. Selectively breeding for big leaves gives you kale or collards. For flower buds, broccoli. For the terminal bud, cabbage.
Wow, all from one ancestor.
All from one Beth. Darwin argued nature does the same thing, just without conscious intent. The environment selects which individuals leave more offspring.
And this needs a lot of time.
Immense amounts of time, which geology, thanks to people like Charles Lyell, was starting to show the Earth actually had.
So survival of the fittest. It's not just about being strongest.
Not at all. Fitness in the evolutionary sense is purely about reproductive success. How many viable offspring do you leave in the next generation compared to others? That's it.
Okay, so Darwin explains the what and the why. But the how at the genetic level,
Yeah.
that came later, right?
Yes, when Darwin's ideas merged with Mendel's work on genetics, that synthesis gave us population genetics.
Right. So for a population geneticist, what is a population?
It's a local group, individuals of the same species living in the same area who can actually interbreed.
And a species.
Generally, a group of such populations that can interbreed with each other in nature, but are reproductively isolated from other groups.
Got it. And the key concept here is the gene pool.
The gene pool, yeah. Think of it as the sum total of all the alleles, all the different versions of genes present in all the individuals of that population.
So each individual is just carrying a small scoop from that pool.
Exactly, a temporary container holding a sample. Evolution, fundamentally, is a change in the frequencies of alleles in that gene pool over time.
And fitness, again, connects back to this pool.
Precisely. An individual with alleles conferring higher fitness contributes more copies of those alleles to the next generation's gene pool. Simple as that.
Okay, but then early on, people wondered, if you have dominant and recessive alleles, why don't the dominant ones just take over?
Yeah. Why do recessive traits stick around? It was a genuine puzzle.
Until Hardy and Weinberg.
Until G.H. Hardy, the mathematician, and G. Weinberg, the physician, independently cracked it in 1908, the Hardy-Weinberg law. What they did was describe a theoretical baseline, a population that's not evolving.
An equilibrium state.
Exactly. They showed mathematically that if certain conditions are met, the frequencies of alleles and genotypes in a population's gene pool will remain constant, generation after generation.
And what are those conditions? They sound pretty strict.
They are. 5 main ones.
But...
First, no new mutations introducing new alleles.
Okay.
Second, no gene flow. The population has to be completely isolated, no individuals moving in or out.
Right.
Third, the population must be very large, infinitely large theoretically, so that random chance events don't cause frequencies to drift.
So genetic drift isn't a factor.
Correct. Fourth, mating has to be completely random. No preference for certain traits.
And fifth?
No natural selection.
Yeah.
All individuals, regardless of their genotype, have equal survival and reproductive rates.
Okay, so basically conditions that almost never happen in the real world.
Pretty much, but that's why it's so incredibly useful. It's like Newton's first law of motion, an object stays at rest or in uniform motion unless acted upon by a force.
So Hardy-Weinberg tells us allele frequencies stay constant unless acted upon by evolutionary forces.
Precisely. When we see allele frequencies changing in a real population, deviating from the Hardy-Weinberg prediction, which we can calculate using their equation, pop plus 2pq plus q plus 1, we know that evolution is happening.
And we can start asking which of those 5 conditions isn't being met? Which force is acting?
Exactly. It gives us a standard for comparison.
So evolution at its core, this microevolution, is that change in genetic structure generation by generation. Let's talk about those forces, the agents of change that push populations away from equilibrium.
Okay, well, we mentioned them implicitly, but let's break them down.
First one, mutations.
Right. Heritable changes in the DNA sequence could be tiny, a single nucleotide swap, or big, like rearranging whole chunks of chromosomes.
And they just happen.
Often spontaneously, yeah. We don't always know the trigger. They happen at a low rate, generally, but they are absolutely fundamental.
What fundamental?
Because they are the ultimate source of all new genetic variation. The raw material upon which all other evolutionary forces act. Without mutation, evolution would eventually grind to a halt.
Like that viper's bug loss example in the book, a mutation causing white flowers instead of blue. That's new variation.
Exactly. That's where novelty comes from.
Okay, agent number 2, gene flow.
Gene flow, simple concept. Alleles moving between populations.
Like individuals migrating. Or pollen blowing in the wind.
Yep, or seeds getting dispersed. When alleles move from one population to another, they can introduce new alleles or change the frequencies of existing ones.
So it kind of mixes populations together.
It does. The main effect of gene flow is to decrease genetic differences between populations, making them more similar. It also tends to increase genetic variation within a population that receives new alleles.
Does it ever work against natural selection?
Oh, frequently. Natural selection might be favoring traits specific to a local environment, making a population unique. Gene flow from other populations with different adaptations can counteract that, kind of diluting the local adaptation.
And for plants, how far does this gene flow reach?
Often, not very far effectively. For many plants, especially those relying on insect pollination or heavy seeds, gene flow drops off sharply with distance. Sometimes just 50 or 100 meters can be a significant barrier.
Interesting. Okay, next agent. Genetic drift. You mentioned this with Hardy-Weinberg.
Right. Genetic drift is all about random chance. It's changes in allele frequencies due purely to random sampling events from one generation to the next.
And it hits small populations harder.
Much harder. Think about it. If you flip a coin 1,000 times, you'll get pretty close to 50% heads, 50% tails. But flick it only 10 times. You could easily get 7 heads and 3 tails just by chance. Same with alleles. In a huge population, random deaths or failures to reproduce might not change overall allele frequencies much. But in a tiny population, losing just one individual who happens to carry a rare allele can drastically change the frequency or even eliminate the allele entirely.
And there are specific scenarios where this is really obvious.
Two classic examples. First, the founder effect.
Founding a new population.
Exactly. A small group breaks off from a larger population and colonizes a new, isolated area, like an island. That small group, just by chance, might have allele frequencies very different from the source population. Their gene pool becomes the foundation for the entire new population.
Like one seed starting a whole new colony?
Precisely. The second is the bottleneck effect.
That's dramatic.
It often is. A population suddenly and drastically shrinks in size due to some random event, a fire, a flood, a disease outbreak, overhunting. The few survivors who make it through the bottleneck are essentially a random sample of the original population. Their gene pool might be very different, with some alleles lost entirely and others overrepresented purely by chance.
So both founder and bottleneck effects lead to a loss of genetic diversity, potentially.
Yes, and significant random shifts in allele frequencies.
Okay, one more agent, non-random mating.
Right. Hardy-Weinberg assumes random mating, but that's often not the case. Individuals might prefer to mate with others nearby, or in plants, they might self-pollinate.
That leads to inbreeding.
It can, yes, mating between close relatives or selfing. Now, inbreeding itself doesn't actually change allele frequencies in the pool.
Oh, what does it change then?
It changes the genotype frequencies. Specifically, it increases the proportion of homozygous individuals with two identical alleles, like WW or WW, and decreases the proportion of heterozygotes, WW, generation after generation.
Which can expose recessive traits, potentially harmful ones.
Exactly. That's often the consequence of inbreeding depression.
Okay, so we have these agents stirring the pot. Mutation, gene flow, drift, non-random mating, and then natural selection acting on the result. It's often thought selection just gets rid of variation, right? Selects the best and eliminates the rest.
That's a common misconception. While selection can reduce variation by favoring one extreme, it can also maintain or even promote it in different ways. And remember, selection doesn't act directly on genes. It acts on the phenotype.
The observable traits, what the organism actually looks and functions like.
Right, and that phenotype isn't just genes, it's the result of genes interacting with the environment throughout the organism's entire life. Think about those Jeffrey pines the book mentions.
Ah, yeah, the ones that are tall and straight in good conditions.
But become stunted and gnarly on a harsh windswept ridge. Same genes, potentially, but the environment drastically alters their expression, their phenotype.
So a trait's fitness isn't fixed. It depends on where the organism lives.
Absolutely critical point. Height might be great in a dense forest competing for light, but a liability in an open, windy area where it costs energy and risks breaking. Context is everything.
And the outcome of all this selection is adaptation.
Adaptation, that's the key result. The term can mean a few things, but biologically, an adaptation is usually understood as a trait or modification of a trait that increases an individual's fitness in its specific environment.
How do we see this playing out?
We see it in response to the physical environment in a few ways. Sometimes it's just developmental plasticity, like we saw with the pine trees, or how a plant might grow different shaped leaves in sun versus shade. That's the environment directly causing phenotypic change without genetic change. Plants are great at this because of their continuous growth.
Okay.
But other times the differences are genetic. We see clines, gradual changes in a trait across an environmental gradient. Maybe plants get shorter as you go up a mountain, or change flowering time with latitude.
A smooth transition.
Yeah. And then you have ecotypes. These are genetically distinct populations within the same species, but they live in sharply different habitats and show clear, often abrupt, phenotypic differences suited to those habitats.
Like that Potentilla glandulosa example, the herb with different types at different elevations in California.
Exactly, from sea level up to the alpine zone. And when you grow them together in a common garden, they keep their distinct characteristics, growth form, physiology. It proves the differences are genetic adaptations, not just plastic responses.
So they're locally adapted populations.
Very much so, like goldenrod adapting differently to shaded versus open fields, or mountain sorrel populations from the Arctic versus alpine environments having different optimal temperatures for photosynthesis.
Fascinating. And adaptation isn't just to the physical world, right?
Yeah.
What about other species?
Great point. That leads to coevolution. This is when two or more species act as major selective forces on each other, evolving in response to each other.
Like flowers and their pollinator.
The classic example. The shape of a flower evolves in relation to the beak shape or body size of its pollinator, and the pollinator evolves behaviors or structures to better access the nectar. Or think about monarch butterflies and milkweed.
The monarchs eat the toxic milkweed.
And evolve tolerance to the toxins, while the milkweed evolves higher toxin levels or other defenses. It's an ongoing evolutionary arms race.
And invasive species kind of show the flip side.
They do. When a plant like kudzu or purple loosestrife arrives in a new place without its usual predators, diseases, or competitors from its native range, it's released from those coevolutionary pressures. It can often thrive and spread rapidly, sometimes adapting very quickly to the new environment.
Okay, this brings us to the big one. The origin of species. Darwin's book title, but he didn't fully explain speciation itself.
Not entirely, no. The mechanisms of how one species actually splits into two or more, that understanding really developed throughout the 20th century. It's the field of speciation.
Defining a species seems like it should be simple, but it isn't.
It's surprisingly tricky. The most common definition, especially for animals, is the biological species concept.
Which says?
That a species is a group of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. The key is reproductive isolation. They can't successfully breed with outsiders.
But that have limits, especially for plants.
It does. It doesn't work for asexual organisms or fossils. And in plants. Well, hybridization between what look like distinct species can be surprisingly common.
Right. You mentioned using the morphological species concept sometimes, just based on looks, on structure.
We often have to, practically speaking. But then you get cases like the London plane tree.
The hybrid.
Yeah, it's a fertile hybrid between two sycamore species that look different and had been separated geographically for maybe 50 million years, yet they can still hybridize successfully.
The lines can be blurry.
They can. Which is why phylogenetic species concepts are also important now. These define species based on their evolutionary history, looking for the smallest distinct group on a phylogenetic tree based on shared characteristics or genetic history.
Okay, so how does speciation happen? How does one gene pool get split?
The most common way, we think, is allopatric speciation. Allo meaning other, patric meaning country.
Geographic separation.
Exactly. A population gets split into two or more geographically isolated groups. Maybe a mountain range rises, a glacier advances, a river changes course, or some individuals just disperse across a barrier.
And once they're separated,
no more gene flow between them. So mutations arise independently in each group. Genetic drift might push them in different directions, especially if the groups are small. And natural selection will likely favor different traits if their environments are even slightly different.
and over time they just diverge genetically.
They accumulate differences. Eventually, they might become so different that even if the geographic barrier disappears and they come back into contact, they can no longer interbreed successfully. They've become reproductively isolated. New species.
And this can happen even over small distances for plants.
Yes, because of their often limited dispersal. Think back to those ecotypes. They represent strong local adaptation, the beginnings of divergence that could lead to speciation if gene flow is restricted enough.
This relates to adaptive radiation, right? Like on islands.
Perfect example. Adaptive radiation is when one ancestral species gives rise to many new species relatively quickly, each adapted to exploit different ecological niches. Islands are famous for this.
Like the Hawaiian lobeliads mentioned in the book.
Incredible example. From likely a single ancestor arriving on the islands, they exploded into about 128 different species. Different growth forms, trees, shrubs, vines, different leaf shapes, different pollinators, birds, moths. They diversified to fill almost every available niche in the Hawaiian forests.
Wow. That's diversification on overdrive.
It really is.
Okay, so that's speciation with geographic barriers. What about without them? Sympatric speciation.
Right, sympatric, same country. Speciation occurring within the same geographic area. This was more controversial for a while, but in plants we have a really clear major mechanism.
Which is?
Polyploidy, having more than two complete sets of chromosomes.
Ah, the extra chromosome sets we talked about.
Exactly. Remember, it can happen in two main ways in sympatry. First, autopolyploidy.
Auto meaning self.
Yep. An error occurs during meiosis. Maybe chromosomes don't separate properly within a single individual of a species. This can produce gametes, pollen, or ovules with double the usual chromosome number. If two such gametes fuse, or if one fuses with a normal gamete and then the chromosomes double later, you can get an offspring with, say, 4 sets of chromosomes, tetraploid, instead of 2 diploid.
Like De Vries's evening primrose and Atherigigus.
Precisely. That tetraploid Anothera was instantly reproductively isolated from its diploid parents because hybrids between them would have an uneven number of chromosome sets and be sterile. A new species formed in one generation.
Okay, and the other type?
Allapolyploidy. Allo meaning other. This involves hybridization between 2 different species.
Which usually results in sterile offspring, right?
Usually, yes, because the chromosomes from the 2 parent species don't pair up properly during meiosis in the hybrid. But if that sterile hybrid undergoes a spontaneous chromosome doubling event, then suddenly every chromosome does have an identical partner to pair with during meiosis.
Making the hybrid fertile.
Exactly, and it's fertile with itself or with other similar allopolyploids, but it's still reproductively isolated from both original parent species. It's a brand new species, combining the genomes of 2 ancestors.
And this is common in plants.
Hugely important in plant evolution. Estimates suggest maybe 30 to 80% of flowering plant species originated this way. Many of our crops are allopolyploids.
Wheat, you mentioned.
Wheat is a classic. Bread wheat is a hexaploid, 6 sets of chromosomes, the result of 2 distinct hybridization and polyploidy events involving 3 different wild grass species over thousands of years. Also happens naturally, like in goat's beard, tragopogon, or the salt marsh grass, Spartina anglica.
That's amazing. Any other ways for sympatric speciation?
There's also recombination speciation. This is rarer or maybe harder to detect. 2 species hybridize, and instead of polyploidy, the hybrid's mixed genome gets rearranged through recombination over several generations. Certain combinations of genes might eventually lead to reproductive isolation, forming a 3rd species distinct from the parents, but with the same chromosome number.
Like the anomalous sunflower.
Yes, Helianthus anomalus. It arose from hybridization between 2 other sunflower species, and scientists have even recreated its origin experimentally.
And even sterile hybrids can sometimes persist.
They can if they can reproduce asexually, vegetatively. Like that horsetail hybrid, Equisetum exferisi, it can't make viable spores, but it spreads readily via underground rhizomes. And then there's epimixis.
Seeds without sex.
Basically, yes. Seeds are formed, but they develop directly from maternal tissue without fertilization. So the offspring are genetically identical clones of the parent. This allows successful hybrid genotypes, or just successful genotypes in general, to propagate widely without needing pollination. Common in grasses like Kentucky bluegrass, dandelions, hawkweeds.
Okay, so that covers how species form. What about the really big picture? New genera, families, orders, macroevolution.
Right, the origin of major groups. The traditional view, stemming from Darwin, is often called the gradualism model.
Meaning big changes are just the result of lots of small changes adding up over long times.
Essentially, yes. Speciation happens, as we discussed. Then one of those new species might accumulate further changes, become different enough to be classified as a new genus. That genus might diversify, leading eventually to a new family, and so on. It's microevolution scaled up over vast geological time, a slow, steady, gradual accumulation of change.
But the fossil record doesn't always look like that.
It often doesn't. What you frequently see in the fossil record are species appearing relatively suddenly, persisting for millions of years, virtually unchanged, a state called stasis, and then disappearing just as suddenly. Smooth, intermediate forms are often rare.
Which led to another idea.
It led Niles Eldredge and Stephen Jay Gould in 1972 to propose the punctuated equilibrium model.
Punctuated, meaning bursts of change.
Exactly. They argued that the gaps in the fossil record aren't just missing data. They might actually reflect how evolution often happens. Their idea is that most significant evolutionary change, the stuff that leads to new species and potentially new forms, happens relatively rapidly, in geological terms, during the speciation event itself.
Especially in those small, isolated populations undergoing allopatric speciation.
Precisely. That's where genetic drift and strong selection can cause rapid divergence. Once a new species is formed and becomes widespread, it might then enter a long period of stasis, equilibrium, with relatively little morphological change.
So the fossil record captures the punctuation, points to rapid speciation, and the long periods of equilibrium in between.
That's the core idea. It's not that gradual change never happens, but that punctuated equilibrium might be a more common pattern for the origin of morphological novelty seen in fossils.
Is this a fight? Like, is Darwin wrong?
Not at all. It's not about whether evolution happens or the mechanisms like natural selection. It's a debate about the tempo and mode of evolution. Does change happen mostly slowly and continuously, or mostly in relatively rapid bursts associated with speciation, followed by stability? It shows evolutionary biology is a dynamic field, still refining our understanding. Darwin probably would have found the debate fascinating.
It really puts things into perspective. Okay, so let's try to recap this journey. We started with Darwin, his voyage, his observations on places like the Galápagos, leading to the revolutionary idea of natural selection driven by variation and differential success.
Right. Then we dove into the genetic basis with population genetics, the concept of the gene pool, and the crucial Hardy-Weinberg law as a baseline for detecting evolutionary change.
Then we unpacked the 5 agents that drive that change away from equilibrium. Mutation as the source of novelty.
Gene flow mixing populations, genetic drift causing random shifts, especially in small populations.
Non-random mating changing genotype frequencies, and the big one, natural selection, shaping organisms to their environment.
Which leads to adaptation. We saw how that plays out physically through plasticity, clines, and distinct ecotypes, and biologically through the intricate dance of coevolution.
And finally, the origin of species, speciation. We talked about defining species' biological, morphological, phylogenetic concepts, and the main modes. Allopatric speciation via geographic isolation.
And sympatric speciation within the same area, especially highlighting the huge role of polyploidy, both auto and allo, and hybridization in plant evolution, plus things like recombination speciation and apomixis.
And we wrapped up by contrasting the macroevolutionary patterns described by gradualism versus punctuated equilibrium. It's quite a comprehensive picture.
It covers the core processes laid out in that chapter, yeah. From the smallest genetic change to the grand sweep of life's diversification.
So as you look around, maybe at the plants in your own backyard or a park, remember all this complex machinery humming beneath the surface. It makes you wonder, what subtle evolutionary pressures are shaping life right now, perhaps in ways we haven't even fully recognized yet, maybe even in response to changes we're making to the planet.
That's a deep thought to end on. The process never really stops.
Thank you for joining us on this exploration of evolution. We really hope this deep dive has given you some valuable insights and maybe some new ways to think about the living world. From the Last Minute Lecture team, thank you for listening.